Click for IB Biology, Principles of Respiration, the Synthesis of ATP. And here we are taking a look at ATP synthase and how it converts the flow of the protons, the hydrogen ions, into ATP. There's a PDF in the description onto which you can write your own notes, and then there's a quiz at the end for you to check your understanding. We left chemiosmotic theory with a high concentration at the point where we had the high concentration of protons in this restricted space. And so the Cristae have this adaptation of the folding of the walls of the Cristae allow these small spaces into which protons can be concentrated by the electron transfer chain. With a high concentration of protons, we then have the capacity for diffusion. So the energy which was in the NADH has now been transferred to a high concentration of protons and a very strong gradient. What was discovered was that these protons flow through the enzyme on the other side called ATP Synthase, and that this enzyme, which is embedded in the wall of the Cristae, has a core pore, so there's a pore, and the protons are flowing through that. And as this occurs, it causes the protein to rotate at the base. This rotational energy is then transferred to this section of the enzyme here, which has active sites for ADP and phosphate. As the rotation completes, the ADP and the phosphate are brought into close proximity, where it is possible for the third phosphate bond to be made to form ATP. So the proton concentration leads to a rotation of the protein, a rotation of the enzymes, a bringing of the substrates into closer proximity so the bond can be made to form ATP. And here on the right hand side we have a diagram extracted from a protein database showing the core section of the base embedded in the membrane, the stalk through which the hydrogen ions are flowing, and up here we have the enzyme head structure component, which must have an active site perhaps here for ADP and another one for phosphate, something of this arrangement. This work was published in 1997 by Boyer and Walker, and another Nobel Prize was awarded for medicine to these characters, the third in the series for in the area of respiration. And here we see an illustration of their proposed model with the rotational subunit of the protein embedded in the membrane, the high concentration of protons here in the Cristae space, and how they flow through the protein structure, emerging into the matrix like so, and how this rotation brings together ADP and P to form AT, a remarkable structure which is so singularly important in aerobic respiration. In further videos, we're going to consider issues such as what happens if there's a shortage of oxygen, or what are the comparisons between respiration in proteins, lipids and carbohydrate. The video link is shown here, the subscription buttons here, and a link to the playlist.